Hello everyone. Today I will show you how to actually find your drug for your study using our tools, actually TRANSFAC disease package, which include various tools which you can use to analyze your gene expression data and epigenomics data together, and to find drug targets, and actually to find new potential drugs which can be useful for your further studies. So let's start. All right, so as you can see, we open a gene expression omnibus entry of one analysis, actually, which was done some time ago, where these authors were studying actually cisplatin resistance, and they did transcriptomics and epigenomics analysis. So first we log in into our system, actually using this account. And we then open Genome Enhancer tool. And the first task is actually to upload the data from that gene expression omnibus. But okay, first we have to create the new project, and it will open the field where we can upload our data. And actually we can upload it from URL directly from gene expression omnibus. We go to this entry, we copy actually the link address, and we go back to Genome Enhancer, put it here in this window, and start uploading. So you see it's now uploaded the raw transcriptomics data. We are starting importing it, and we have to mention that it's transcriptomic. Now it is imported, it's done. Now we need to import epigenomics data. In this case it was actually DNA methylation. And DNA methylation is a complex stuff, so in this study they did it using actually special platform. And now we take the file. Actually, it was several rounds of creating cisplatin resistant cells. We take the latest round of those resistant cells. We upload the data actually to our computer. Here is the file. As you can see, the format of the file is pretty complicated. So let's just use ChatGPT to understand what this file means. So I just upload it into ChatGPT and ask a question. Please read this structure and convert it actually into an easy Excel table. Also, it checks which genome build it was used, and it took information from this data file, and actually it's put it in the Excel table. Now we open this Excel table, we can see, and in fact we need to actually look at this data and export it in a BED format. This is standard format how genomic coordinates are actually stored. So you see this format should have special fields like name and score, and also this strand. So we put this necessary information in this Excel file and export this as a BED file. Basically, as you can see now, but we delete also some bad lines which has no scores. And now we uploading this BED file into the system. So now we have to define the build, it's HG19, and define its epigenomics data. So now we have transcriptomics and epigenomics data in the entry, in the project. Now we put all the files which we are going to work with into this gray box of Genome Enhancer, where we put transcriptomics and DNA methylation data. Next, we need to define actually the conditions. So we have two conditions: cisplatin resistant cells, which were developed in this experiment, and cisplatin sensitive cells. So we name those conditions accordingly, resistant and sensitive, and we drag and drop those files which are corresponding to the resistant cells, and those which correspond to the sensitive cells. And then we put also DNA methylation data in the resistant cells. Now, next step, we define which disease we are actually analyzing, ovarian neoplasm, and we compare resistant cells versus sensitive cells. So we start the analysis. It will take some time. So in the meantime, we can, read the books. For instance, very important book about gene regulation in eukaryotes. You can read it in the meantime when the analysis is done. It will take maybe a couple of hours because it has inside certain algorithms which actually try to find differentially expressed genes and analyze promoters of those genes. Those upstream analysis pipeline which is done here. So, but what we'll do, we'll look at these ready results in the of this study, and we have among the examples these ready results. So the genome enhancer generates such an output. You can see here this. It found the most important targets. You see here the set of data which was analyzed. First, what the system does, it actually defines the differentially expressed genes. It's also builds such a heat map to see whether these two conditions really can be discriminated. Then finding those upregulated genes and mapping them on gene ontology to understand that actually, and also to the pathways, as you can see, like G1 and cell phase are very abundant there. And also the same is done for downregulated genes. Right, but this is still the downstream analysis, which tells you what these genes are doing. Still, we have to find the drug targets. We have to understand how these genes are regulated, and for that, this next analysis is done. So where we actually start analyzing enhancers, basically those regions of in the 5' areas of the genes where the differential methylation was observed. And we found that these genes actually are regulated in the resistant cells by specific cluster of binding sites in the promoter of those genes. Why it's important? Because now we actually know which transcription factors are actually important for misregulating those genes in the resistant cells. Well, those cells are happy because those genes are regulated, but now we know these transcription factors, which are shown here in these tables. And next is actually to go further upstream from the transcription factors and identify master regulators. As you can see on this map, the system has done this. So we see this map of master regulators. These are pink molecules here, and the transcription factors and their target genes are at the bottom of that diagram. And among master regulators, you see, for instance, TGF-beta receptor and TGF-beta, signaling, which seems to be very important here in the resistant cells. All right, so you see here that, for instance, SMAD2 transcription factors, which are well known in that pathway. So, and again, those molecules are selected as the most promising drug targets. The system also actually makes this kind of schema, how these things are regulated. At the end, it selects those drugs which would work best against those selected master regulators, drug targets. And as you can see, first of all, we see those well-known drugs which are already known for ovarian neoplasm with a high score. But what is important for our discussion today are new drugs, or repurposing drugs, which were perhaps never used yet in such kind of setup, but maybe they were used in different types of cancer, like this compound, for instance. And our tool actually identifies that with a quite high score, drug score, this compound can be very interesting. Another compound which actually also might be promising, again, it was used in leukemia and HIV studies, but in this case we can see that this compound can target the drug targets which we revealed. And most interesting, we actually can also identify completely new compounds, which are mentioned in drug bank just as a interesting molecule, and which doesn't have even a proper name as a drug, but it seems this molecule, according to our chemoinformatic calculation, can target those important master regulators which we found in our study. So this way, as you can see, we can actually go quickly from your just raw data, from gene expression data and DNA methylation data. It will combine it, it will find differentially expressed genes, and then it actually in the 5' regions and regulatory regions of those genes, it will identify where the DNA methylation differentially happened in the resistant cells, for instance, in this case, and then doing all the upstream analysis from this data, basically understanding which transcription factors are involved in regulating the expression of those genes in resistant cells, and then which master regulators are involved in regulating activity of those transcription factors, pinpointing to the potential drug targets, and even further steps to see which drugs which are already on the market available for ovarian cancer can be combined with cisplatin in this case to tackle this resistance problem, or even go further to find new compounds just from the active chemical structures, which give a first hint, and as in our project, which was just finished, where we actually found a new compound and patented it. We actually started from some promising chemical structure and then adapted it further with a different chemistry and found actually very potent new anti-cancer drug. Thank you very much for your attention, and I hope you can apply our tools for your drug discovery studies. Goodbye.